Hi everyone. So thank you so much for joining us today. We are happy to organize a new workshop which will serve as an introduction to Ensembl REST API. This workshop is also part of the R for Bioinfo series that you have launched since September 2020. And today we feel really honored to be hosting Dr. Aleena Mushtaq and Dr. Michal Szpakwho. So let me just introduce myself. I'm Muna Belaid. So I'm working as a business intelligence consultant at Prime Analytics. And I'm a organizer at the Ladies Tunis. So Hidiat Nani, who's also the organizer, at Ladies Tunis, and I are responsible for the organization of this meetup, and really are happy to see you here with us. So Dr. Aleena Mushtaq background is in biomedical and molecular sciences. She completed her PhD in 2020 at the University of East Anglia, UK. And so for her doctorate, she studied the dynamic relationship of host and microbiota in the gut-liver axis in response to Western diets. She joined EMBL-EBI as an Ensembl Outreach Officer in 2021 and delivers training on ensemble resources. We are really happy to have you today. Dr. Michal Szpakwho took a degree in biology at the University of Warsaw, where he studied the ancient DNA. So he subsequently moved to bioinformatics and explored human genetic variation across different ethnicities in his master's degree research at the University of Virginia. So his doctoral and postdoctoral work focused on positive selection and genetic adaptations in humans. He also joined EMBL-EBI as an Ensembl Outreach Officer in 2019 and continues to deliver Ensembl training worldwide. Really wonderful career, so really congratulations for your work. So before moving on, let me just introduce to you our ladies Tunis through this video.
Hello and welcome to this workshop session. Before starting the workshop, we'll introduce you to the Our Ladies Global and Our Ladies Tunis initiative. The R community suffers from an underrepresentation of minority genders in every role and area of participation, whether as leaders, package developers, conference speakers, conference participants, educators, or users. As a diversity initiative, the mission of Our Ladies is to achieve proportionate representation by encouraging, inspiring, and empowering people of genders currently underrepresented in the R community and to facilitate individual and collective progress worldwide. Gabriela de Queiroz founded Our Ladies on October 1, 2012. Our Ladies Global was born and the grant was awarded in September 2016. Since then Our Ladies has grown to 170 chapters in 44 countries and 39,000 members. Our Ladies Tunis is part of Our Ladies initiative and was created in May 2020 by women working as data scientists and biostatisticians and bioinformaticians. Our goal is to create an R community in Tunis and empower underrepresented genders in the R community. This is the core team of Our Ladies Tunis. Come and join our community on social media. This presentation was made with R Markdown. Thank you for watching this video and enjoy our workshop.
Thank you all for your attention. The screen is yours, our dear trainers.
Okay. Thanks very much, Mona, for that nice introduction, and really nice to meet everyone. Right, so we're going to be speaking about how to access Ensembl's data programmatically. So I'm going to share my screen now and turn off my camera for a while. Okay, so here is the course agenda. So during the courses, we're going to talk about the REST API and how to query the REST API with the URL. And we're also going to have a little general introduction to Ensembl. And then later on, Michal is going to talk about fetching REST endpoints programmatically, and then also talk about loads of different additional topics. Before I move on, I just wanted to say that if you have any questions, do unmute yourself or write them on the chat. Michal and I both will be monitoring the chat. Or if my screen isn't zoomed in enough, you can't see it, then please unmute yourself and definitely tell me. So you can find the course materials at training.ensembl.org, and this is where you will find a copy of this presentation, but also Jupyter notebooks. So we have notebooks that are available in R, and you can, so we also have human example or plant example. So let me just jump out of the presentation and I will just show you the page. So I'm on training.ensembl.org. And over here, if you focus on this.
Sorry, Aleena, we can't see the website.
Okay, weird. Thanks.
Yeah, good now.
All right, great, thank you. So I'm on training.ensembl.org, and over here you can find materials for the Our Ladies training course. So I'm just going to click here and go to Our Ladies REST API course. And over here, you'll be able to find the slides from the presentation. We have the Jupyter notebooks, and I'll expand on this in a bit. And then we also have the Living Doc. So Living Doc is a Google document. And over here, you can ask questions, and you can ask questions anonymously. And then Michal and I will try our best to answer them. And then it's also really a nice place to go back, so you can save it, and then you can go back to the questions and the answers for references. And then we have the Jupyter Notebook. So we have this with Google Colab, and then we also have it with MyBinder. For the Jupyter notebooks, we have human examples, and then we also have some plant examples if you're working for, if you're researchers in plants, for example. So let's click on Jupyter Notebook, Human R. So this is an interactive notebook, and we host them, as I mentioned, by either Google Colab or MyBinder, and I'm on the Google Colab bit, and you'll see that I'm signed in here. So we would recommend that you use Google Colab, as it allows you to save and make changes to the notebooks. You can also save your code and save all of your answers. To use Google Colab, you have to have a free Google account. And if for some reason you don't have a Google account, then you can use MyBinder. The only drawback with MyBinder is that it won't let you save your answers and your code. So this is what the Jupyter Notebook looks like. So you have a little introduction here, and then you also have different exercises. You also have this run button here, so you can run your code. And you'll see the output underneath it. And then for some exercises, you have a box to write your own code. And then underneath the exercises, you have the answers, which are hidden. So if you get stuck or you want to work backwards, so you can follow the answers and reach your solution that way. Right, so I'm going to jump out of this and back to my presentation. And I think when Michal covers his topics, he will expand on the Jupyter notebooks a bit more. Michal, could you let me know if you can see my slide?
Yes, it's all good.
Great. Okay, so let's talk about Ensembl Features. Ensembl is a comprehensive database. It's composed of different features, including genome assemblies and gene builds for over 300 vertebrates. We also have some comparative genomic analysis, which we calculate in-house. This includes whole genome alignments and gene trees. We also have regulatory builds. So these are regulatory features that are annotated from experimental data in human and mouse. And we have variation data and a web tool. So this is variant effect predictor tool, which allows you to annotate your own variation data. We also allow you to visualize your own data. So you can plot your data against the genome in the browser. And then we also have Biomart, which is our online data export tool, allowing you to export Ensembl data in bulk. We also support programmatic access to our data via our APIs. So we have Perl API and also REST API, which is language agnostic. And then finally, Ensembl is completely open source. It's open access and it's free to use. So Ensembl is dedicated to vertebrates. We have about 300 vertebrates, over 300 vertebrates in Ensembl, but if you look closely, you will also find some non-vertebrate model organisms like yeast, fly, and worm for comparative purposes. Then we also have some mouse strains and agricultural animal breeds. We have a sister site, which is called Ensembl Genomes, and this site is dedicated to non-vertebrates, and it's split by taxa. So there's a separate site for bacteria, protists, fungi, and metazoan species, also plants. And these are all color-coded, but they follow the same logic and the same organization as the main site. Here we have a table which compares Ensembl and Ensembl Genomes. So Ensembl is older and Ensembl Genomes was released in 2009. The main difference between the two sites is that we annotate all of the vertebrates in Ensembl ourselves, while the non-vertebrate site in Ensembl Genomes is annotated in collaboration with scientific communities. So we input information from external sources. So for example, for Drosophila, we input it from Flybase. However, there's only one REST server to rule them all. Sometimes if you're working with non-vertebrates, you have to specify some additional optional parameters, but we will talk about this later on. So we have dedicated servers to both the newest human genome assembly, which is GRCH38. This assembly has fewer gaps and it gets updated regularly. So it is the most updated human genome assembly. Then we have a separate REST server for GRCH37. And this is the previous human genome assembly. It's still widely used. So this is used mostly by the clinical community. However, it has more gaps and data is rarely updated. And this can be found at grch37.rest.ensembl.org. So Ensembl works on a release cycle. So Ensembl data is not static. We update our data regularly, which means that every 3 months we have a new Ensembl version. And the current Ensembl version is 104, which was released in May. There's a lot of behind the scenes work going on during this time. So we input new genome assemblies, we update our gene sets, update our software, and run new analysis on this data. So we, for example, we do new comparative analysis, we update variation and regulation data, and then finally, we package all of this up to display it in the genome browser. So because of these updates, it might also result in a new REST endpoint being added, or maybe sometimes there are changes to the existing endpoints, in which case we have the REST archives. And these start from release 87. So the REST archives for are for the current human genome assembly, so for GRCH38 only, and they're kept for up to 5 years. And this matches the Ensembl web archives. To use these archives, you will have to use a slightly modified server. So you will need to add the prefix, which indicates the archive that you're interested in. So for example, it's E for Ensembl, and then the Ensembl release number, which is 87, and then .rest.ensembl.org. Our REST API is modeled around biology. It follows the parent-daughter relationship. So higher up, we have DNA, which is translated into pre-mRNA, which is then spliced by removing introns to give a mature mRNA. And then the coding portion of the mRNA is translated to give protein. The primary features in Ensembl are genes, transcripts, and exons. Pretty much anything which can be mapped to the genome is a feature. And then a gene is a set of alternatively spliced transcripts, and a transcript is a set of exons. We also have translations, but those are not features in Ensembl, as they do not map to the genome directly. They're mapped to the genome via transcripts. So they're a product of a transcript and they're not stored in the database. These are computed on the fly by the transcript object, and translations are defined by CDS, so coding sequences. I would also like to remind you that not all exons are coding. There are non-coding exons, which are called UTRs, untranslated regions. And it's also important to note that not all transcripts have translations. There are many non-coding transcripts, like RNA transcripts, which do not produce protein. All of our features have a defined location on the genome. So every feature has a defined start and end position. And it's important that the start and the end of a feature is always defined on a forward strand. So this means that feature start and end might not reflect the transcription start and end. And this is because the start coordinate always has to be smaller than the end coordinate in Ensembl. So we have two examples here. So we have the forward stranded gene and the reverse stranded gene, and the start and the end are always defined on a forward strand, irrespective of a gene orientation. So for a forward stranded gene, the feature start would match the transcription start, and the feature end would match the transcription end. But for the reverse stranded gene, the feature start would actually match the transcription end. I hope this makes sense. There are some exercises that tackle this, so it will make sense a bit better later on. REST API stands for Representational State Transfer, and it describes how one system can communicate with another. So imagine a client, for example, so your computer communicating with our server over HTTPS using this language agnostic method to access some data or service. So for example, if your computer sends a URL to our server specifying the data that you want, you can see that we have this endpoint here called data I want, which is obviously made up. So our server will interpret this request and return requested data in a specific format. Ensembl REST API is language agnostic. This means that it provides access to Ensembl data independent of any specific programming language. So you can use many different languages to access our data using our REST API. However, it only provides a fraction of the Perl API functionality. However, it seems to be sufficient for most people that use REST API. You can find all of the documentation on rest.ensemble.org. Just a little bit about what Ensembl REST API is and is not. So Ensembl REST API provides HTTP access to Ensembl's data. And it's a stable service, which means it doesn't change very much. It's limited by network latency. This means that the network speed is going to affect it. It's read-only. This means that you're not allowed to edit our database. You only have the read access, and it's also versioned in archives, as I mentioned before. It's not hypermedia as the engine of application state or fully RESTful. If this doesn't mean anything to you, it's okay. It's for the super techie people because there is a discussion online about what fully RESTful really means. And if we are completely there yet, but again, don't worry too much about it. There's no mirrors. So if you are familiar with Ensembl, you might know that there's the option for the website to work with different mirrors for different parts of the world. So we have US East, West, and Asia, but we only have one REST server for programmatic access, which is based here in the UK. Finally, it's not a hugely efficient data mining solution. So it's not as fast as direct as you would have access with MySQL or FTP side. And it also provides incomplete coverage of what's available there in the database. So on my slide, I have a description of what an endpoint is, but in more human terms, endpoint is a particular output that you can get given a particular input. So it's a function that interacts with our database. You can find the full documentation of all the endpoints at rest.ensemble.org. And this documentation lists all the endpoints grouped by function. So they're grouped by the output, and you can find all of the required parameters for each endpoint and optional parameters, and also example codes on how to use the endpoints in different programming languages. So I'm going to jump out of my presentation now, and we'll go to the documentation, which is here. So I'm on the page rest.ensemble.org. And over here, you'll notice that we're on the endpoints section. And on the page, we have different REST API endpoints, and these are categorized. So we have archive, we have comparative genomics, cross-references, EQTL, and loads more. So there are loads of endpoints on this page. So the best way to find the endpoint that you want or you're interested in would be by searching for it specifically. And you can do this by using Control F, or if you're on Mac, then you can just use Apple F. So for example, I'm going to search for gene tree, which is here. And I can find the gene tree on the page. So I found my gene tree endpoint. And now I want to bring your focus to a specific endpoint, which is lookup. And over here you have get and post, and these are the different types of HTTP method. We're going to focus on get for now, and then Michal will speak to you about the post method. So we have this endpoint here that is get lookup /id /:id, and I will talk about this a bit more. And the description for it is that you will use it to find the species and database for a single identifier, which could be a gene, transcript, or a protein. So I'm going to click on this hyperlink. And now I have more information about this get endpoint. So we have our HTTP method first, which is get, and then we have our endpoint. So we have lookup/ID/:ID. So anything that comes after a colon is our required parameter. If I scroll down a bit, so over here, we have our different parameters. So we have required parameters, which is, in this case, ID, and this is a string type. And then we have the description for it. And then we also have an example for it. So the required parameter is your, so this needs to be replaced with your input, this bit that comes after a colon. Then you also have the optional parameters, and these can be used to extend or modify your output. So you have loads of different optional parameters here. For example, you have the phenotype optional parameter, and this is a Boolean type. And you can use this to give you more information about associated phenotypes. So as I mentioned, this is a Boolean type. So by default, it's set to 0, which means null, but you can turn it on by typing 1. And then on the right-hand side, at the top of the page, we also have resource information. So first, we have the method, which is get, and then you have the response formats. So these are the different representations that you want your data in. So over here, you have a list of JSON, XML, and JSON-P. You don't need to worry so much about the content type. We will expand on it a bit later on as well. So now, if I scroll down the page, we have some example requests. So if I bring your focus to this hyperlink, we have our nicely constructed URL here. So you have the lookup/ID, which is your endpoint. Then you have your required parameters. So we have the Ensembl gene stable ID here. And then we have a question mark, and then followed by the question mark, we have our optional parameter, which is the content type. And this is set to application JSON. In the box underneath, you have the example output. So if you were to make a call with this endpoint, this is the output that you will see. And then very helpfully, we also have the different examples of scripts in different languages. So we have...
Sorry for interrupting you.
Sure.
Could we please increase the size of the screen?
Yes, definitely. Thank you.
Thank you.
Better? Thanks.
Yes. Yeah, thank you.
Right. So as I mentioned that, firstly, we have the example output, and then you have the script in different languages. So we have Perl, Python, Ruby, Java, and also R. So I'm going to click on this hyperlink here to make a REST call and to see the output in the browser. Okay, so let's focus on the URL first. So firstly, we have rest.ensembl.org, which is our server. Then we have the lookup ID, our endpoint, followed by our required parameter. So this is our gene stable ID. And then we have the content type, which is the optional parameter, followed by the question mark here. And this is set to application JSON. If I want, I can also change my Ensembl gene stable ID. So I'm just going to change the last digit and hit enter. And this is now going to update my results output. For my results, I have my gene name, the type of object, the strand, so the gene falls on the forward strand, and then I also have some more information about it. For example, the description of the gene, the start position, and the end position. So this is shown really nicely over here in Firefox, because Firefox is a more JSON-friendly browser. In other browsers, you might see your output like this, which is not very readable, but it is correct still. So let's go back to JSON. Another thing that I wanted to show you was, so I'm going to go back to the our get lookup ID documentations page. And I wanted to show you that you also have the option to add the optional parameters. So for example, over here, we have an expand optional parameter. This is also a Boolean type. And what this will do is it will expand the search to include any connected features. So if the object is a gene, then you also have information about its transcripts, translations, and exons. Information will be returned as well. So let's focus on this URL now. And I'm going to open this URL in the browser. Okay, so for this URL, we have our server, which is rest.ensemble.org. Then we have our endpoint lookup/ID/ our Ensembl genes table ID. And then after the question mark, we have our optional parameters. So first, we have our content type, so which is set to application JSON, and then separated by a semicolon. So if you want additional optional parameters, you can separate them by semicolon. We also have the optional parameter expand, which is turned on because it's 1. So in the output, you will see the information about the gene, but then you also have information about the transcript, and you have information on the exon as well. Okay, so I think this is all on the get lookup. I'm going to go back to my presentation now. So I've showed you how what endpoint documentation looks like. Again, the required parameters are indicated by :ID. So :ID in the documentation, so that's something which you have to replace with your actual input. And then there is a list of optional parameters. These can be applied so that you can add them to your URL, and they follow this format. So there is a parameter name, which equals the desired option, and then they can be added after the question mark. So if you want to include multiple option parameters, you have to separate them by a semicolon. And then you also have the descriptions on the documentation page. And then you also get a sample code. And then there's also the different scripts. So making a REST call in the browser. This is something that we've already covered. Is the easiest way of making REST calls. All you have to do is put your URL in the browser, and this can be used as a quick lookup. And you can you can test your URLs to see whether they work, whether you've included the correct parameters, and what the output looks like. You probably won't be using the browser call very often, as you're most likely, it's more likely that you would want your output in a file rather than in the browser. However, we have an example of a browser call which you could be using quite often as it's handy. So this is a ping endpoint, which confirms that you have a connection to the database. So here we have the URL of this particular call. So we're starting with the REST server. We have the HTTP / rest.ensemble.org, then the endpoint, and then after the question mark, we have the optional parameters. This is probably not needed. So if we run this and the return is a 1, it means that everything is good and that you're connected. And if the return is anything else other than 1, then it means that you probably don't have a connection to the database and that most likely perhaps our servers are down. And then over here, we have an example of a request for a gene using the gene stable ID. So this is exactly what I've demonstrated before in the on the Firefox. But just to recap, so we have our constructed URL here. We will start by the server, so which is rest.ensemble.org/endpoint, which is lookup/ID/ followed by the required parameter, which is the Ensembl gene stable ID. And then after the question mark, you have your optional parameters. And in this case, we're specifying that the response format is application JSON, and we also have what the return JSON will look like. So this is a structural data type, which is composed of keys and value data pairs. Status codes. So you should never assume that your request has run successfully. The server uses HTTP status code to signal the request outcome. So for example, if your client makes a request of something which doesn't exist, so perhaps you've made a typo in your endpoint, or you're using an endpoint that doesn't exist, or you're requesting data of a certain ID which doesn't exist, depending on the problem, the server is going to return a status code. And in this case, 404 means that the URL was badly formatted. So basically this endpoint doesn't exist. And then we also have the other different status codes. So for example, 200 means that your request was successful, and everything else other than 200 would mean that something went wrong. 400, for example, means that your parameter was wrong. So perhaps you asked for a gene stable ID, which is not found in the database. 403 means you probably made too many requests. So we have some limits. We have a quota of number of requests that you can make per second. So if you exceed that, you might get blocked temporarily, and then you will get the 403 status code. 404 means that your request was badly formatted. So perhaps there was a typo in your URL. And there are some more. So everything that starts with a 5, so for example, 503 means that most likely it was our fault. So it means that perhaps the server is down. You can find a full list of all status codes in our GitHub, which is in the link on my slide here below. Okay, so now it's time for exercises. The first exercise, you're asked to go to the documentation. You will need to find an endpoint which you can use to look up information about a gene using its symbol. You're then asked to build a URL to find information about a specific gene. And here we have the SPN gene in human. And I think finally you have to, you're asked to expand your results to include information about transcripts. And we did this before in the demonstration. And to give you a little clue, you will probably have to use optional parameters for this. So I think we will we'll say about 10 to 15 minutes for the exercises, and then we'll come back and I will probably go through the exercises with you. So please go to the documentations and also to your Jupyter notebooks. This is on the training page that I shared previously. There's some introduction section in the Jupyter notebooks, and then you have your exercises, which are followed by answers. I would like to remind you that in this case, you're not running a code in the notebook, as we're only asking you to make browser calls. So just please create those URLs, test them in your browser to see if they work, and then type your answer in the Jupyter notebook. So we'll go through the answer in 10-15 minutes. Okay, so it's almost 15 minutes now, so I think it's time to move on. If you've struggled with the exercise, don't worry about it. We'll go through it together now. So I am on rest.ensembl.org, and we're told that we want to look up a gene by its symbol. And when I look at this lookup section here, you can see that you have the get and post methods. So we will be talking about post methods later on, but for now we're going to focus on the get method. So we've covered lookup ID earlier, and that's looking up information about a feature using the feature ID. However, in this exercise, we're interested in looking up a gene using its symbol. You can see that we have another lookup endpoint which retrieves information for a feature using its symbol. So I'm going to click here to have a look at its documentation. So here we have a nicely, so we have a constructed URL. So to start with, we have our get method. Then we have lookup /symbol/ our required parameter, because it's followed by a colon. So we have to specify the species / the next required parameter is the symbol. So ensemble stable IDs are unambiguous, which means that every feature has its unique stable ID. However, gene names are not unique. And because they could be exactly the same gene name in human or mouse, therefore, if you're trying to look up a gene using its symbol, you have to specify which species you're interested in. And the species can be provided in either Latin name or the common name. And then we have the symbol, which is the for the gene. So if I scroll down, we have an example URL. And I've mentioned this loads of times, but let's just go through it again. So we have our lookup /symbol/ Homo sapiens. So this is our first required parameter. We've specified our species here / our gene symbol. So this is BRCA2. And then followed by the question mark, we have our optional parameter, which is the content type set to applications JSON. So let's open this hyperlink. So in the URL, we first have our server. We have our lookup endpoint. This is set to BRCA2. but the gene that we're interested in is ESPN. So I'm just going to change this, and I'm going to change this to ESPN, and I'm going to hit enter. So now our results output shows us information about the ESPN gene. We have its stable ID, we have its description, and we have its biotypes. It's a protein coding gene, and other information about it. So this part answers the question, the part 1 of the exercise. So which lookup endpoint you will be using. It's the one with the species and symbol. And we have also used the ESPN gene symbol to find out more information about it. Then the third point is exercise asks us to expand this output to include transcript information. So let's go back to our endpoints page. And over here, we have another nicely constructed URL. And again, we have the endpoint here, followed by the required parameter for species. Then we have the gene symbol. And then after the question mark, we have our optional parameter. So first we have the content type set to application, and then separated by the semicolon, we have our other optional parameter, which is expand. And this is turned on because it's a Boolean type by specifying 1. You can find more information about this optional parameter here. So you have the name, which is expand, the type of the optional parameter, and some description about it. And it also tells you that by default, it's set to null. So let's open this hyperlink. So you'll see that because we also have in our optional parameters, we have content types set to application JSON, and then separated by semicolon, we have our additional optional parameter expand, which is turned on. And we have information about the ESPN gene and more information on its transcript and exons. And this completes exercise 1. I'm going to stop sharing my screen now and pass on to Michal.
Thank you very much, Aleena. Let me just share my screen now. Lovely. I hope everyone can see my screen now. Before we start, I just wanted to show you how you can copy the notebooks to your own Google Drive. I think Aleena might have already mentioned that, but just in case, let's go through it again, because now we're going to be moving to scripting. So we're going to be making a REST API calls using scripts. So if you go to the Ensembl training page, your course event, you'll find those links to Google Colab with our Jupyter notebooks. And if you open either plant or human examples, as Aleena showed you earlier, you will find our copy of the Jupyter notebook, which you can run, you can play with, but you cannot modify and change. So if you wanted to make your own copy, just click on File, and then save a copy in Drive. And in that way, you will save a copy of this notebook into your own Google Drive. You have to first sign into your Google account. You can see I'm signed in over here. Great stuff. So let's now go to my presentation. So the whole beauty about the REST API is that it allows you to make calls using scripts. Okay, so it's a very powerful and flexible tool, because often you're not going to be wanting to make your calls in the browser, as Aleena showed you, because you're not going to be interested in having all of this output and all of that information in web pages, but I would rather make calls in your scripts and then save the output in files and then modify them further or extract bits of information or use them as an input in other scripts. So basically scripting out REST API calls allows you to extract specific bits of data from your REST call. So yet again, often you get lots and lots of information and you might be interested only in specific data point. It allows you to output the results in your preferred format, so you can manipulate and modify the output. You can specify whether you want the output as FASTA format, for example, or simple text, or so on and so forth. You can also print your output modified customly. You can then link together calls for more complicated queries. So you can chain endpoints together. So for example, you can use output of one call as an input for another. And you can also integrate your queries into a larger pipeline. So you can basically link up many different databases and tools to build those little pipelines. And we're going to be talking about all of that now. So as Aleena mentioned, REST APIs are designed to be accessed using any programming language. So that means that the REST API is language agnostic. So it's independent of any specific programming language. And calls can be made and decoded within any script. And we have examples in Python, Perl, R, but also Ruby and some others. But if there are any specific programming language that you're interested in and we don't have those examples online, please let us know and we might consider to provide more examples. So throughout this course, we're going to be using different R libraries. So as you probably already know, libraries are, they kind of make our life easier because they have lots of stuff predefined in them so that we don't have to code many different functions from scratch. And today we'll be using the HTTR library to make requests in R within your script. And then we will be using JSON Lite package to decode the JSONs. And here I'm showing you how you can load your R libraries. All of the libraries and everything is pretty much set up in the notebook, so you don't have to worry about installing anything. But obviously, if you'd like to make calls in your own environment, in your own machine, you have to configure it accordingly. So you have to set up and install all of the packages which are needed for dependencies. So this is what Aleena showed you, what we've been doing so far. We've been requesting Ensembl data using URLs in the browser. So we're making those browser calls. And here we have an example of the URL and the output, but you can do exactly the same within a script. So let's talk about it now. To do so, we're going to be using this get function from the HTTR library. So all we have to do, we have to sort of feed in our URL into this function to make a request within a script. And here we have a couple of hints how it can be done. So let's go over the code in more details. So first of all, it's a good practice to define your objects and variables beforehand. So that, for example, if you want to change your variable later on, you don't have to be changing it all over your script. You can just change it where it was defined for the first time, so just change it in one place. So what we're doing in this bit of code, so we're kind of creating our little script, and we're starting with loading our libraries. So we first load the HTTR library, and then we're loading JSON library, and then we're starting to define our objects. Okay, so we're first making a string of the server, which again, we're going to be using those objects multiple times, potentially later on. So we're defining our server object, and it's going to be a string, which is indicated by this quotation mark. And if you're not familiar with strings, obviously there are many different object types in R and other programming languages, and string is basically a sequence of characters. So we now know that our server will be stored under the server object. We're going to have our server is going to be stored under the server object, and then we're defining another object which we call extension basically here, where we're going to have the rest of our URL, so basically our endpoint. And then we are making our call within a script using this get function from the HTTR library, which was predefined, so we don't have to worry about it. So what we are doing, we are pasting our server and extension strings separated by nothing. So we're basically feeding in our URL. And then you can notice that actually we're not specifying the content type as a optional parameter in the URL. Instead, we're specifying the content type in the request header. And we're going to be talking about the headers later on. So basically the request headers are sort of like a metadata. The request metadata, sort of like different options. And there are many different standard options, including content type and accept. So far, Aleena's been using content type, and the accept headers are similar. So they're both used to specify the format output, and we're going to be talking about how they differ in a little bit. So instead of specifying the application JSON content type in the URL, we're specifying it in the header under accept. And then we're storing our response in another object, which we call R for response, and then we're printing our response. Another important thing is the error handling. So as Aleena already mentioned, you should never assume that a response will run correctly. So it's good to have this sort of built-in mechanism to check the response code returned by the server in case something goes wrong. And here we have an example of how to do that. So we are already familiar with the first bit. So we already covered that. We import our libraries. We're defining our objects, which we're then going to use to build our URL within the script. And we are making our request using the get method. And then we're printing our response. And then there's another function which is already predefined in the JSON Lite package, I believe, one of those packages. So again, it's a ready function that we don't have to worry too much about. So basically what this function does, it checks the status code returned for you. And then if it's different from a 200, so as you already know, as you remember, a 200 means everything is great, everything went fine. But if the response code is different from 200, that means something went wrong. So basically the stop for status function checks the status code of our response. And then if it's different from 200, it raises an exception and it shows you what went wrong. So now I wanted to jump out of my presentation and actually show you that in action. So let's jump out of my slides and go to your, no, not this, go to your Jupyter Notebooks. I hope you can see it fine. Please do let me know if you wanted me to zoom in. So let's move to the second section, making requests with R. So this is exactly what we just covered. So if you see, if I run this block of code, so what we're doing here, we basically, we are making a call in the script. So we're making a request and then we're printing the request. So let's run this code to see what's going to happen. It might give you this warning message saying that this notebook hasn't been authorized by Google, basically hasn't been produced by Google, it's been done by us, but nothing to worry about. I'm going to run it anyway. And here we can see this is our response. So this response is not decoded. Therefore, you can't really see this information that you saw in the browser. I'm going to tell you in a minute how you can decode the JSON to see the output. So if you don't decode the response, you're going to see there's a bit of information about the response, like the date, the content type, size of it, and the status code. So you can see the status code is 200. That's great. That's exactly what we want. And then if you look at the second bit of code, you can see we have one new line which says stop for status. This is our error handling. So what it does, again, it basically says, if something goes wrong, show me what it was. So if I run it, you can see I can't see any output. It's because nothing went wrong. So I can't see any exceptions being raised. But as soon as I make a mistake, so let's make a typo in my gene ID. So if I delete 10 and I'm going to run it again, you can see I got an error message saying that it was a bad request and the error code is 400. So basically this optional parameter is incorrect. So this object doesn't exist. But for example, let's make a different mistake. So let's say I make a typo in the extension. So let's say I'm going to add another P and let me run it again. And now you can see the status code is different. So now we've got 404, which means that the request was badly formatted and actually this endpoint doesn't exist. So in that way, you can kind of learn something about your errors. So if something goes wrong, you might get some hints. So it's great for debugging your scripts. And also just to mention, a great start of debugging will be to use the ping a request that Aleena showed you. So it will be first good to check whether actually you're connected to the database, because sometimes you might be getting some errors because of a connectivity issues. All right, great stuff. So let's go back to my slides now. Let me have a look if there are any questions. I can't see any questions. That's great. But if you have any questions, please feel free to type them in the living document, or you can also unmute yourself and ask the questions out loud. Let's now talk about the headers. So HTTP allows serving of different representations of the resource based on the client preferences. So the data might be returned as text or JSON, application JSON. So basically the structured data data type, which can be interpreted by a programming language. So it's seen as the kind of objects that you can manipulate and extract. The data can be returned as image and so on and so forth. So content type and accept headers are kind of used by the server and client to negotiate what formats they're going to communicate with. So accept is basically more of a wish where you specify what type of data you're ready to accept, you'd like to receive, while content type is basically says what type of content, what format has been returned by the server. So what was the actual format? So in a way, they're kind of similar. The difference is what side of the request those are located. So let me actually jump out of my presentation and go to the Firefox to show you that in the browser. So if I go to Firefox, you can see this here. We have a call that I made in the browser. It's one of the calls that Aleena has showed you. So you can use any browser call to see what I'm about to show you now. So here we have our JSON friendly output. And Firefox is great because it's very easy to look up the headers. If you're using different browsers, you might need to dig in the HTML code to find those headers. But Firefox has this another tab with the header information. So I'm just going to click here. And here you can see those options that I was talking about. So headers, again, these are basically the a request and response options, okay? So you can see here under the request headers, so at the side of the request that we have this accept, and we haven't specified accept in this request. You can see we only provided content type in the URL, but we haven't mentioned accept anywhere. So we're sort of ready to accept all sorts of different formats. And then at the response side of the request of this call, we have content type category, and you can see the content type is application JSON, which is exactly what we requested in the URL. So I hope that's kind of makes sense, but don't worry too much about it. It's just this kind of nitty-gritty details. So let's go back to my slides now. And as Aleena mentioned, you can find the available format types in the upper right corner and our documentation pages. And you can find the full list on our GitHub page, which is really handy because actually this documentation page only lists you types of accepted output formats, but it doesn't say you how to actually, what's the specification, how to specify them in your script. So for example, you can see that application JSON is one of the allowed formats here, but it's not telling you that to use it, you actually have to say application/JSON. So you can find the full specification in GitHub. So let's now talk about decoding the response. So throughout this course, most of the time, we're going to be dealing with a JSON as our output format. So this is the structured data format that we're going to be talking about in more details in a second. And most programming languages have ready parsers, so have this built-in parsers that can return the JSON, can interpret it for you. And this is how you decode JSON in R. So remember, R is our response object. So we can decode it using this content function. So we say content, and then in the brackets, we give the name of our response, and then we're returning is a text, and we can also specify the encoding. So just basically Unicode encoding, 8-bit encoding. And then we're storing our decoded JSON under the decoded object, and then we're printing the decoded JSON using Prettify. So basically, Prettify allows you to print the JSON response in this nice human readable format, similar to this Firefox display. And here we have an example of a JSON. You already saw it in the browser. So yet again, JSON is this data structure, okay? So it's basically like a dictionary in Python or a data frame. So it's composed of these keys and values data pairs, okay? So you can see that the JSON is indicated by those square brackets, which again indicate dictionary, and then its main content is those keys and values data pairs. So you can see here we have, for example, a biotype key and its value is protein coding, or we have a display name, which is a gene name, and its value is BRAF gene. And sometimes those, let me see, there was a question, I think, how to know what to put in the encoding argument. Just put what I showed you. Don't worry too much about it. You can just use this a Unicode 8-bit. And sometimes those JSONs get complicated and difficult. They can get quite massive and horrendous with many this nested data frames. And it might be difficult to locate your desired value in this big structure. So sometimes you might first have to print the entire response and then have a look to see where your desired values are located. And you might need to cycle through a list of nested data frames to get to your desired data points. And there are some tools which allow you to, which are basically might make your life easier, and they allow you to interpret those big difficult JSONs for you. Let me just see if I'm not going ahead. Okay, I'm going ahead. We're going to be talking about that later. That's all right. So I'm going to show you how to kind of decode these JSONs if you don't have this nice Firefox display. And let's now talk about helper functions. So it's very advisable and it's very good practice to use functions in your scripts. And again, functions make your life easier. So sometimes if you are, for example, working with many different genes and you wanted to make multiple calls of using different gene names, or for example, you might be using different endpoints, and they're all structured similarly. So if you wanted to make multiple requests, you would end up repeating same blocks of code repeatedly. And instead of doing that, you can define a function and you can chuck in all sorts of bits of code, blocks of code into the function body, which you're going to be using repeatedly later on. So the function kind of serves you as a template you can be using later on. So you can, for example, define a function and put the request making in it. You can put the error handling, decoding of the JSON, and returning and printing of your response. And then you can just use this function whenever you need. And here we have an example how to build such a function in R. So what we're doing first, we are giving our function a name. So here we're calling it fetch endpoint. So we're defining our function. And then we are giving it 3 parameters. So the first parameter would be server. The second parameter would be request. And please don't get confused, as so far we've been calling this parameter extension, but it really doesn't matter how you're going to call your parameters or objects. It's entirely up to you. And then our third parameter would be the content type. Here we have some a comment line. And then within the function body, we are making our call. So again, we're declaring the R object. We're using the get method to make a call. So we're pasting our parameters, so our server and our requests separated by nothing. And then we are specifying the content type under accept in the request header. Then we have our error handling. And then we check for we're basically deciding what output format we want to print depending on the requested content type. So if the requested content type was application JSON, then we want to return JSON, and otherwise we're returning the response as text. So if you want JSON, we have to use this from JSON or to JSON function, which basically it's again, it's predefined in the packages, and it allows you to convert between JSON data and R objects, so between JSON and data frames. So what we're doing here, we are converting the JSON to R's data frame using exactly the same content function and the same encoding. So now we have time for your second exercises of to date. We're going to write a script to look up the gene called ESPN in human and print the results in Json. But before we move to the exercises, I wanted to show you one more thing in your Jupyter notebooks. Let me just have a look. There is one question. Is it structured opposite to Python? So, Json is structured pretty much the same in Python and R. So, it's also this data structure with those keys and values data pairs. So let me just jump out of my presentation and go to Jupyter Notebooks. Not here. Here. I just wanted to show you an example script and kind of point out differences between the function parameters and then your global variables. So here we have exactly what I showed you on the slides. So here we have our little script. And what we're doing, we're loading our libraries. We're then defining our functions. So we're calling it fetch endpoint, and we're giving it 3 parameters. And again, you can name those parameters however you like. And then we are telling the function what to do with the parameters inside the function body, so that the function is going to know that the parameters are given in this exact order, and then it's going to know what to do with them, how to apply them exactly. So we then making our request. We have our error handling, and then we are returning the relevant content type. And then outside of the function body, we are actually defining our global variables. So we're actually saying what the actual server is, what the endpoint is, and what the content type is, okay? So we're defining our objects for server and extension and content type, and then we are making the call. So we're calling our function by using its name. So we're saying fetch endpoint, which is our function's name, and we're feeding in our objects or our variables in the exact order. So we first give it our server, we tell it what the server is, what the endpoint is, and what the content type is. And then if you want, you can also store the response in another object, which we call here getGene. And the last thing we do, we're printing our JSON using Prettify. And again, if we want to print it as a JSON, we have to sort of, again, convert from the data frame to JSON using this toJSON function. And then we're giving it the response object. Great stuff. So now we have some time for your second exercises. So you are asked to write a simple script to look up the gene called ESPN in human. And print the results in JSON. Please, you don't have to, please feel free to copy and paste anything from those boxes. So we give you an example code, which you're more than welcome to start using in your own scripts. Please don't feel like you have to code everything from scratch. Just copy paste this code and then modify it accordingly to solve the exercise 2. So go to the documentation, find the relevant endpoint, which can look up the gene using its name, and then make a script call. And let's say we have about 15 minutes for this, and then we're going to review in 15 minutes to see where we are. If you have any questions, if you're getting stuck, if you're completely lost, please shout out. There's a really good question on the living document, which Margarita asked. I actually wanted to bring your attention to it. So this content type parameter, which Aleena showed you earlier while you were making the browser calls, is actually unusual parameter, because the normal and standard way of specifying content type or accept is in the headers. So our REST API is kind of unique because we allow you to specify the content type in the URL itself as well. So I would say that the content type parameter is quite unusual. So if you're scripting, you should specify your content type in the headers, but all of the other optional parameters like expand or all sorts of parameters that we allow for should go to the URL. So you can include them in your endpoint variable object. So, you know, your extension object specifies the string of the endpoint, and you can add your optional parameters there after the question mark. So you can have their extension equals 1. But sometimes also you might want to, if you wanted to, it's entirely up to you, because there are different ways of doing it, and there's no wrong or right way of doing it. So for example, if there are some parameters that you know you're going to be changing a lot, for example, if you're working with many different genes, you might want to define an object for the required parameter like gene name. So for example, you will have gene name object, which you can be changing frequently. And then you could be using this gene name object to build your URL as well, sort of adding this gene name or gene ID to the rest of your endpoint. And I'm going to show you that later on. So how are we doing, everyone? I can see 3 hands up. I'm not entirely sure whether that's from the previous exercises or whether you just raise your hands now. So we already spent 15 minutes on this exercise. I think I'm going to be moving slowly to the model answer. So again, don't worry if you struggled with it. Hopefully this walkthrough now will clarify some things. And then I want to quickly introduce one more concept before we all leave today. So let's now go to the Jupyter Notebook to have a look at the answer. So here we have the model answer, which is hidden by default, but you can reveal it by clicking on the reveal option. And this is not much different from what we just saw in the little demonstration before or on the slide. The only thing was to find the relevant endpoint. And actually, we already used that endpoint earlier. So Aleena showed you how you can look up the gene information using the gene name. And we know that in that case, we have to specify the species as well, as gene names are ambiguous. So they could be exactly the same gene names in human and mouse. So you have to specify which species you're actually interested in. And you already looked at the documentation pages and you know how to add optional parameter, how you can expand your output to include the additional information. Although I don't think we're asked to expand. No, I think we're just only asked to look up the gene using its name. So yet again, let's just go over the code to refresh what we just covered. We're importing our packages. We're defining our function. We're giving it 3 parameters. We then we have the request making in the function body. We're using the get method. We're pasting our server and extension, so our endpoint object separated by nothing. We're specifying the content type in the accept header. We're checking for an error, and then depending on the requested content type, we're returning our output as either JSON or text. And then outside of the function, we actually get to define our server, our endpoint, and so on and so forth. So what we're doing here is actually what I just mentioned earlier. So we are first defining our gene name object. You don't have to do it like that if you don't want to. It's just one example of doing it. So again, if I think I might be working with many different genes of different names, it might be handy, it might be useful for me to have this object so that I can be changing the gene name only here at this point in my code. But you can as well add the gene name to your the rest of your endpoint if you wanted to. We're then defining objects for our server. So we're using the same server as always, rest.ensemble.org. We then defining our content type as application JSON. And for example, here we could also define another object for the request or extension if you wish. But you can see whoever wrote this code actually decided to hard code the extension in the request making. But again, it's entirely up to you and your coding practices, your coding habits, how you want to address that. So then we are making the request. Oh, sorry, actually, here we have exactly what we're doing. We're defining our extension, so our endpoint here. So here we have extension, and what we're doing, because we defined the gene name separately, we are now concatenating. So we're pasting the endpoint and the gene name separated by nothing, and we're also adding the question mark. You see, so now we're building the extension over here. Alternatively, you could declare the extension with the optional parameter in one go if you wanted to. And then we are making our call. So we are using our function name, we're feeding it all of those objects we just defined. But also, if you wanted to, you can specify your parameters just over here without predefining them earlier. And then we're storing the response under get lookup, and then we're printing our JSON using prettify. So if I run it, that should give me my response as JSON. So you can see it's structured. This JSON format is indicated by this curly bracket, and we have those value keys, data pairs. Okay, great stuff. So before we go, we still have 10 minutes, I believe. So I just quickly wanted to show you one concept. So since JSON is this structured data type, so data frame in R, you can pull out a single data point using the key name. So for example, the value of display name, which is basically gene name, is BRAF. And if I wanted to only print a specific data point instead of all of this information, because yet again, often, what you're getting in your response is lots and lots of information, but you might be only interested in a very specific data point. You can easily do it in R by first specifying the data frame, the entire data frame name, and then you can use this dollar symbol to specify which key you're interested in. So you have to specify the key name to extract its value. And again, we are storing it under another object, which is symbol. And that's important, especially when we're dealing with this really nasty, horrendous nested data frames, because sometimes you might have this more complicated structures like here. So here we have a list, which is indicated by the square brackets. And on that list, we have 2 data frames, so 2 dictionaries, each composed of this keys and value and data pair. So this is basically output of the overlap endpoint. So what it does, it takes as an input genomic coordinates. So this is our parameter and then it outputs features that overlap this genetic region. And you can see that the feature type is set to gene. So what we're doing, we're basically outputting genes that overlap this genomic region, and each gene is a data frame or dictionary on its own. So you can see we have 2 genes overlapping this genomic region. So that's why we have this list of genes. And if these JSONs are big and horrendous and difficult to read, you can use one of those tools. These are external tools. We don't develop them. All you have to do is basically copy your output JSON, paste into one of those tools, and it's going to interpret it for you, and it's going to break it down into paths. So you can then click on the object you're interested in, and it's going to tell you where in this structure your object is located. And I just wanted to show you an example. So let me go to my browser again. Here we have an output from another endpoint. So this is phenotype term endpoint. So what we're doing, we are printing here genetic variants associated with a specific phenotype, in this case, coffee consumption in human. And we're going to be using this endpoint in one of our next exercises. So here we have a list of genetic variants associated with coffee consumption. And every variant on that list is a data frame with another nested data frame. So you can see this is one genetic variant. You can find its genomic location, the variant ID. So here we have the RS number, but then under attributes, you can see another nested data frame. So another nested dictionary. And there's some more information there. And you can see on that list, we have quite a lot of variants. So there is 205 variants because we started from 0. So what I can do, if your browser displays this JSON like that, which is absolutely horrendous and unreadable, you can copy this JSON and you can go to one of those tools, like for example, JSON Pathfinder, and then I can paste my JSON here. And then what it does, it breaks it down into paths. So what I'm getting is this nice display similar to the Firefox. And then if you want to click, if you point at any specific object item on that list, it's going to build, it's going to give you a path to this value in this data structure. So for example, let's click on this and it tells me that this specific PubMed ID is found under external references, under attributes of the item 28th. So now we have a set of exercises, which I will ask you to try on your own as a homework. So we have 3 different exercises on that list. So first is quite straightforward. So what we ask is to write a script to look up the gene called ESPN in human and print the stable ID of this gene. So it's pretty much exactly what we've done earlier. Please just copy paste this code and just add next step to extract your desired value. And then we'll start the next class from reviewing those exercises. So I will hand over to our host now. Do we have any closing remarks for today?
Thank you so much for all of you for attending and for your attention. Really great job. Would like really to thank you for this informative workshop and very good explanation, in fact. Yes, and I like how there were exercises, so the session was interactive. That's really good. Thank you.
Thank you very much.
So if there is another question, please don't hesitate to ask. Otherwise, I want to let you know that the second part will be held in 1 week, June 11, at the same time. So you are welcome to join us. I will share information about the registration right now in the chat. So you have shared the event so in the meetup page already. Don't miss the opportunity to be attending next time. Okay. So I think it's okay. You can close. Thank you all for attending and have a nice weekend.
Thank you for joining, everyone. See you next week.
Thanks.
Thank you. Goodbye.